Hello, everyone. Thank you for joining us today. And as you're all just coming in on screen, um, I'll just let you know that I'm very excited to have so many people that have been registered for today's webinar, not just from Australia, but from around the world. It's just fabulous to have this level of interest in ecosystem. Um, and to start the webinar today, and in keeping with the spirit of reconciliation, we at TERN acknowledge the Jagera people and the Turrbal people as the traditional custodians of Meanjin, which is Brisbane, where the lands on which our TERN head office is located. We pay our respects to the traditional custodians throughout Australia, New Zealand, and all nations. We're proud to be building on and partnering with our first environmental scientists, benefiting from their profound connections to land, water, biodiversity, culture. Just getting my slides to work on. And as such, we pay our respects to their elders, past and present and emerging, and acknowledge the important role that Aboriginal and Torres Strait people continue to play within our community. So again, thank you for joining us today from all over the place, all over Australia, all over the world. It's very gratifying. And for those that haven't met me before, my name is Beryl Morris, and I'm the director of TERN, Australia's Ecosystem Observatory. And just in case you don't know about TERN, our role is to advance ecosystem science through our long-term monitoring of the environment and our preservation of the data so that research can happen and we can contribute to the sustainable management of Australia's environment. TERN is not really a simple entity. We're actually a collaboration of so many different partners, the universities across Australia and CSIRO, and we also have global contributors to what we're doing. We're enabled by a grant from the Australian government, which is called NCRIS, and our work is a result of all of those collaborative partnerships. And you can find out a lot more about us online. So first of all, before I talk about the speakers, just make sure that you submit your questions as we go along in the webinar today via the Q&A button if you can, not the chat button. Use the chat button for any technical difficulties that you're having with sound and picture, and we'll try to pick those up, but use the Q&A button so that our speakers can have a look and maybe answer some of them as we go, but we'll be answering questions at the end. I'm so pleased as the beginning of 2025's webinars to have such a stunning lineup. We have Mark Westby from Macquarie University. Now, Mark was on the original advisory board for TERN, so we owe so much to Mark. Good to see you, Mark. We have Hannah Carl, and we're very pleased that Hannah is able to join us. She's working at Western Sydney University, but Hannah's also running a national plot network of rainforest trees. And Ben Sparrow has been with TERN a very long time, running our surveillance platform, making sure that we have laid out across Australia a plot network and transects. I'm not going to read out all of the bios for them because we sent them out to you, and it's probably one of the reasons why you joined us today. I, in fact, want to switch off my video and my audio and ask Mark Westerby to start telling us some of the things that he would like to contribute to our topic of site experiments plus long-term monitoring and what does it all add up to. And thank you, Mark. Over to you.
Many thanks, Beryl, and really appreciate the opportunity. Thanks for all of you out there for coming. So I'd like to begin with this proposition that in the whole of ecology, or at least the whole of terrestrial ecology, there is one thing which is both seriously important and we really don't understand very well. It's sort of the top of the list of those two things, and that is how plant species range boundaries work along, especially temperature gradients, along all kinds of environmental gradients, but temperature gradients are the most important for this purpose. So here are some data from species at higher rainfall in eastern Australia. And what's happened here is that we're starting from furthest from the equator and highest up mountains and proceeding towards the equator or downhill in slices of 1 degree C. And the blue dots on the graphs are how many species you gain for each degree C. That is, you've crossed their cold boundary. And the red dots are how many species you've lost for each degree C. That is, you've crossed their warm boundary. And if you just focus on the right-hand side of the picture, that is the same numbers expressed as a proportion of all the species in that temperature slice. And roughly speaking, you average about 20% of species gained and 14% of species lost per degree C warmer. You gain more than you lose because you're getting more species as you go towards the equator. So pretty brisk turnover. Now, by no means are all of those boundaries due to temperature tolerances directly, meaning that, for example, species just die in heat waves further towards the equator. And to identify what the actual mechanisms are, it's been known for a long time that we need field experiments. And I should specify that when I say experiment in ecology, that doesn't just mean taking measurements in the field. It means applying treatments such as removing competitors or excluding herbivores and comparing what happens in those treatments with controls. Here's a diagram that you can find in almost any textbook since about the 1970s of Joe Connell's famous barnacle experiments on a rocky shore in Scotland, actually. He was American, but he did his PhD in Scotland. And you have one genus, Balanus, the sort of blue-gray one occurring lower on the shore, and Thamellus, the brownish one occurring higher in the shore. And you can see that the boundary between them is well correlated with tide height. And the experiments done consist of moving each genus within their own range of distribution for a control and into the other range of distribution for the experimental treatment, and doing that either transplanting into the middle of a crowd or transplanting while clearing the immediate neighborhood of competitors. And when you do that, you find that indeed the upper boundary of Balanus, the blue-gray one, is set by desiccation tolerance. So it's correlated with what's with the physical environment in the way that you might expect. But the lower boundary of Thamellus does fine if you transplant it further down the tidal height, provided you protect it from competition by Balanus. So its lower boundary is not set by the tide height as such, it's set by the competition. And this general idea, as I say, has been in every second-year ecology textbook for a long time. And yet, amazingly, really, we still have relatively few experiments beyond the boundaries of distributions. Why we don't is a fascinating digression into the history of ecology, but I won't digress for the purposes of this talk. Here is a summary of the total numbers of experiments we had as of a couple of years ago. And I'll just point to one thing in this third row here, that at warm boundaries, competition is a significant factor about a third of the time at a minimum. But the more general point is we don't have enough experiments to know how to predict which mechanisms are operating at which boundaries. And it matters because the outcomes depend. Now, this is a complex slide. I'm sorry, I don't expect you to read all the text on the right, which is about outcomes and mechanisms. Just look at the cartoons on the left. So in the top row of cartoons, that's the initial situation with a cooler species in green and a warmer species in dotted. And the 4 different cartoons below that are what you expect to happen depending on the mechanisms controlling the boundaries. And I'm just making the point that depending on what the mechanisms are, pretty different things can happen as climate moves. Sorry, this is all assuming we're sliding climate to the left. That is, temperature zones are moving from warmer towards cooler. So when I was first thinking about all this, I thought, oh, well, maybe we can somehow inspire an collaborative international network that would somehow do lots of coordinated experiments, transplanting species beyond their warm boundaries, with and without suppression of competition, pathogens, and so forth. But sadly, this hasn't happened and seems unlikely. But my point for this talk is that actually now we sort of have a similar experiment being done the other way around. That is to say, we are transplanting temperature zones across species. This is as you all know about climate change. It's an enormous unplanned experiment. It wasn't solicited by any scientists, and it costs far more than any granting agency would ever have provided. But for better or worse, temperature zones are being transplanted across species by about 1.5 degrees, mostly so far, and clearly it'll be more over the next few decades. So that gives us an opportunity from the climate zones being over the next 50 years, let's say, from the climate zones being transplanted across species distributions. Now, obviously, we will learn something as that goes on from monitoring plots, such as, for example, TERN's, OzPlots, and SuperSites, and other people's monitoring plots. But we stand to learn much more efficiently if that monitoring can be combined with experimental manipulation. Now, there are lots of issues in how to design experimental manipulation to sort of run in parallel with monitoring schemes. They discussed at more length in the paper that's cited there, but I'll just mention a couple of them. Maybe first and most obviously, the experiments need to run over decades, partly because it takes decades for the climate change to happen, but also because it takes time for the population dynamics to work itself through, for colonization to happen, for seedlings to grow up into saplings and adult plants, and for competition to work itself out, and so forth. On the plus side, given that these are fairly slow processes, the research doesn't necessarily demand very frequent visits. These are not plots that need to be monitored weekly or anything like that. There's an institutional problem in the sense that certainly in Australia and probably worldwide, research grant systems mostly will fund you for 3 years of intensive work, not for 3 or 4 decades of much less intensive work. And that's a problem we need to find our way around if we can. A second design issue is that I think we need experimental plots in lots of places, as many as we can manage. And that's basically because we expect the movement of plant populations, well, probably any organism populations, to be very spatially patchy. So, 1 or 2 plots are not going to capture it very successfully. In other words, I personally at least would advocate for simple experiments at many locations, more so than complex experiments at few locations. And I'm saying that because I kind of know how this works in the sociology of science. You sit down to try to design an experiment and you think, okay, well, we have to provide, we have to remove the competition or not remove the competition. We have to maybe provide colonists and as seedlings or as seeds or maybe as both. But really, we ought to do it sort of with and without excluding herbivores as well. Maybe we ought to try clearing some kinds of competition like the grass, but not the trees, and the reverse. Maybe we should be experimenting with providing rhizobia and mycorrhizae with colonists or not. So in the hands of an enthusiastic set of scientists, you can easily blow out to 6 or 7 factors, which is 64 treatments or 128 treatments, even when each factor only has 2 levels. And if you let it do that, then you'll finish up with maybe, 2 or 3 such experiments in the continent. As I said before, for me, I think I would pitch for keeping it as simple as possible, and doing as many in different places as possible. And I think I would pitch for competition and for providing or not providing dispersal colonization as the top priorities. Where would you put experiments? My guess would be the top priority should be places where ecosystem structure changes. In other words, where colonization from a warmer environment might produce a substantial change in the physical structure of the ecosystem. For example, right across northern Australia, there are zones where woodland with grass underneath gives way to grassland. And that would be one sort of high priority zone, but there are certainly others. So in summary then, thanks for your time. Take-home messages are two, I suppose. One is that we have very inadequate understanding about mechanisms controlling species boundaries, and that's important because they actually make a big difference to outcomes under climate change. And the plus side that I'm pitching is that we have the opportunity for a new type of experiment with temperatures now being transplanted across species distributions, but we need experimental manipulations, not just monitoring, in order to learn efficiently from all of that. Great. Thanks very much.
All right. Thanks, Mark, and thanks, Beryl. Yeah, I'm really pleased to be here talking with you today. So thanks to everyone for attending. It's a really interesting point of discussion about the combination of site experiments with long-term observations. As Beryl said, I'm a postdoctoral researcher based at the Hawkesbury Institute for the Environment. And my work centers on understanding climate risk and sensitivity of ecosystems and the impacts of climate change on ecosystems. And my focus really has been forest ecosystems, a lot of work in tropical forests and some work now also in eucalyptus woodlands. So I have done some intensive climate manipulation type experiments and some work in long-term observational plots and with long-term forest inventory data. My work hasn't explicitly combined the two, but what I'm going to do today is offer some reflections on both approaches and where the sort of gaps are left when the two aren't married together. Before I do, as Beryl mentioned, I coordinate this network of plots called the Queensland Permanent Rainforest Plots Network, or QPRP. This is a new coalition of very old rainforest research sites in Australia, and it's supported by a multi-institutional network of researchers. So if you want to know more about these plots, about the data, and what we can do together, then please go to the website or reach out to us on Instagram or Twitter. So what I'm going to do today is give you 3 short sort of chapters to the talk that examine different components of my work reflecting on this combination of long-term monitoring with experimental data. The first of these I wanted to talk about is with regards to cyclone impacts, and this couldn't be more timely this week, obviously, with Cyclone Alfred forecast to hit the coast of New South Wales and Queensland. We can't really do experiments where we damage forests in the way that cyclones do. It'd be hard to get the approvals for that, but we can use long-term monitoring plots sort of as a natural experiment, provided that they're geographically distributed and have been impacted by cyclones to varying degrees. So we know the climate is changing. These graphs here show increasing temperature, vapor pressure deficit, VPD, which is a measure of atmospheric dryness, and climate water deficit, increasing anomalies in all of these variables through time in Australian tropical forest regions. And at the same time, cyclone intensities are forecast to increase in northeastern Australia under climate change. Now we can see some of the impacts of cyclones on these forests. We see large landslides, big trees get knocked over. The destruction is quite visually obvious, but there's been very little quantification of these impacts. And there's still kind of limited understanding about how long these impacts last and how well forests recover from them. So what does this increase in cyclone intensity under climate change mean for our tropical forests? The first thing that some of my work has been able to show is that cyclones suppress the carbon sink capacity of woody biomass in Australian tropical forests. So the carbon sink capacity is the balance of how much carbon that woody biomass is absorbing through growth or releasing when trees die. The graph here has the effect of cyclones on the x-axis and different measures of forest dynamics on the y-axis. So things to the right of the dotted line increase after a cyclone, and things to the left-hand side decrease after a cyclone. I don't expect you to take all of that in, but I'll just point out a couple of key things. So cyclones kill trees. They increase the mortality rate, not just immediately when cyclones happen, but for some time afterwards also. And because there are less trees per hectare, the sort of plot level or per hectare biomass gains that we get are suppressed following cyclones. So if we add these gains and losses together, we get a suppression of per hectare carbon sink capacity for the woody biomass in that forest. Now that's really interesting. Also because we've been able to show that those impacts persist for quite a long time. So on the x-axis here, we have time since cyclone. On the y-axis, we have just one of those measures from the right-hand plot. So we've got instantaneous mortality. The red dots on the plot are censuses where censuses that followed a cyclone. It could be a year after a cyclone, up to 30 years after a cyclone. The blue violin plot on the left-hand side is the distribution of mortality in plots that were not affected by cyclones. So this is a sort of background mortality rate. And we can see after cyclones, we look at that model, we get elevated mortality, and that mortality rate remains elevated for at least 10 years following cyclones. Therefore, we would expect a suppression of carbon sink capacity for that woody biomass to persist for some time. We're moving on to this second piece I wanted to share today. This is again thinking about geographically distributed long-term monitoring plots in combination with climate change as a sort of natural experiment. Now, I've been really interested in how climate change is impacting Australian forests in general, but in my PhD, my focus was primarily on tropical forests. And if we look to other parts of the world, we know that the carbon sink capacity of the Amazon tropical forest is declining. I'm not talking about like the whole basin, the whole Amazon basin as a whole. I'm talking about the capacity for 1 hectare of intact forest to absorb carbon. If we look at similar evidence for Africa, it's a little bit less clear whether those forests are changing through time in their carbon sink capacity. And then some work from my colleague David Bauman showed that there's been a doubling of tree mortality rates in the Australian tropics since 1985. And that suggests a pretty considerable perturbation, potentially, of the carbon sink in our forests. So we looked into this. The graph on the right-hand side here has year on the x-axis from 1971 through to 2019. And it's mirrored at the top with the corresponding carbon dioxide levels for each year. And then the y-axis, the top panel here, is net change in above ground woody biomass. So this is our carbon sink capacity of woody biomass. And the bottom panel is biomass gains from stem growth and recruitment into the greater than 10 centimeter diameter size class, which for anyone who's worked with forest inventory data will make sense. This is a common threshold that's used. So what we see is that prior to about the year 2000, woody biomass in these forests was acting on average as a net carbon sink. And that since the year 2000, the average behaviour has been of net release of carbon from woody biomass. Now, this happens because mortality has increased, which means there's more biomass loss from the forests than there was 40 years ago, but also because biomass gains from stem growth and recruitment haven't increased in the ways that are predicted by Earth system models, and indeed in the way that we see for forest in the Amazon basin, for example. And we've been able to show that this change has been driven by increasing temperature extremes, vapor pressure deficit extremes, which is that measure of atmospheric dryness, and climate water deficit. So this is driven by climate change. But there are some important discrepancies in play here, and site experiments would really help to bridge the gaps. So first of all, as I said, we see this evidence for a long-term decline in the carbon sink capacity of woody biomass from the inventory data. But Earth system models broadly predict increasing sink capacity of woody biomass in tropical forests. And actually, remote sensing data for NDVI for the east coast of Australia shows canopy greening over the past 2 to 3 decades. So what's going on here? One story for woody biomass, perhaps a different story for the canopy. And site experiments, I think, can help. They could help us answer questions like, what is the response of photosynthesis to each of these important climate drivers if we control for the others? Is the carbon from increased photosynthesis going somewhere else, as has been shown for free air carbon enrichment experiment in eucalyptus woodland in Western Sydney? So that extra carbon going underground instead of to woody growth. Are there growth limitations due to nutrient availability or other things? And there's some really interesting work that's been done previously that we can look to for guidance about how to design these sorts of experiments. Okay, so the final chapter, I wanted to look at the flip side. This looks like I've said intensive experiments on climate change impacts. This is using controlled climate facilities. And I wanted to reflect primarily on the challenges that arise here in lieu of having these experiments paired with long-term monitoring plots. So my current work is focused in Western Sydney, also known as the Cumberland Plain. And this is a really interesting space to work in. It's a highly fragmented landscape that's been really deeply impacted by land clearing and land conversion over the years. But it's still really biodiverse. It hosts something around 1,500 plant species, as well as wild emu and platypus and koala populations. So there's still a lot of natural heritage in this landscape. At the same time, climate change is impacting this part of the world, and everyone's probably heard about how hot Western Sydney's forecast to get. We're looking at changes of 3 to 5 degrees potentially by 2090 under business as usual emissions scenarios. So we need to think about how to deal with the enormous floral diversity in Western Sydney in the context of climate risk. So to do this, we think about trying to summarise floral diversity in a few key ways. We think about different functional forms within the ecosystem. We think about climate exposure, both historical and present day. And we think about the traits or characteristics of species that might make them more sensitive or resilient to climate. And then we do big experiments where we try to capture as much floral diversity as possible. You end up with really beautiful glass houses that are really difficult to manage. So we're attempting here to capture as much of the sort of ecosystem as we can with these controlled experiments. And when we do, just to give some insight, we're tending to run with between 10 and 32 species of the 1,500 odd species that occur in this region, sometimes including contrasting climates of the source populations. And generally, we're looking at temperature by water factorial designs with 2 to 4 or 2 to 6 levels of temperature and water to really understand the form of the response to these different climate drivers. What that lands you is experiments with between 1,200 and 2,500 plants, or tens of thousands of seeds. So this becomes, as Mark was sort of saying, really challenging really quickly. But we feel that it's important to try and capture as much of this complexity, particularly in terms of that floral diversity, as we can. And just a couple of examples of insights here we have on the x-axis 4 different temperatures, on the y-axis stomatal conductance, a measure of how much the plants are transpiring. And here are 3 different eucalyptus species. So they're grown at 4 different temperatures, either under well-watered wet conditions or water-limited dry conditions. And we can see that different species are more or less dependent, their temperature response is more or less dependent on water availability. We can also think about functional types, as I mentioned, so trees, shrubs, grasses, and forbs. This is looking at the difference in biomass growth that you get under dry versus wet conditions in the top panel, or warm versus ambient cool conditions in the bottom panel. And what we see, for example, here is that shrubs and grasses tend to be more consistently sensitive than trees and forbs do. So capturing some of these broad stroke elements of functional diversity can be quite insightful as to ecosystem responses. But to scale these findings, we really need to seize opportunities to synthesize these findings from, with other work, for example, from field-based multi-season monitoring of physiology, experiments like EucFace, which is the free air carbon enrichment experiment in eucalyptus woodland in Western Sydney, and also new plot networks that are being established in the region. So to conclude, my 2 cents and my strong feeling here is that the value of legacy long-term monitoring plots really cannot be overstated. We can answer so many interesting questions with them. They represent critical national and, in fact, international research infrastructure, and they should be funded to reflect that. And long-term monitoring plots, they have so much capacity to be leveraged more than they are currently, and indeed to be paired with low impact and or close by site experiments. For example, if we're to establish experiments next to long-term monitoring plots so we don't impact the long-term legacy of those sites directly, we compare insights on physiology or from manipulation experiments with long-term growth data for the same species at the same site. I think I'll leave it there. Thanks very much, and I will pass to, I can stop sharing. There we go. We'll pass to Ben when you're ready.
Yeah, hi everyone. Thanks for the opportunity to chat to you today. When Beryl asked me to present, I thought about my long journey with TERN. Beryl mentioned I was involved since pretty much the beginning. And sorry, the thing that occurred to me was what we're seeing on our 15-year journey so far is the speed of environmental change is increasing. And along with the speed of that change, we have increased access to new ways to assess that change, and our need for information to support management in those areas is also increasing massively. And experimental manipulations provide an option to obtain that information much more rapidly. You're all familiar with various versions of this graph. This is the temperature increase against a 1950 to 1980 long-term average. So I'm going to skip over this graph, but just look at the part of that graph that's occurred along our TERN journey. And you can see there the very beginning of our journey back in the mid-2000s through to 2010 or so. Change had slowed down a little bit, but you can see then over the years and up to current, that seemingly is ramping up significantly. And concurrent with that, we've designed a method, we've implemented that, we've got that truly operational. But then out to the right of the graph, you'll see we've more recently got access to new technologies and new techniques that have largely revolutionized our way of assessing change rapidly. And so I wanted to touch on that just a little bit in my presentation. So we want to understand that change. I think the manipulation experiments often boil down to wanting to accelerate change so that we can understand that change and react to that change more rapidly than simply waiting for that to play out in a natural environment. And so how do we do that? Well, I think there's 3 ways we can do that. We have access to a new suite of tools that we never had access to before, and certainly TERN will help give you access to some of those. There's the concept of substitution or proxies. So there's the space for time concept, particularly in relation to transects, that I'll touch on just in a few slides. And then there's manipulation experiments tying in with a long-term monitoring program. So I'll cover off on those 3 points quickly. This is a graph showing how Google Earth engines revolutionized our ability to detect change in our environment. So what this graph does, and credit to Krishna, I think he might even be online, a postdoc based at UTAS, is he's taken a clip of one of our super sites here and then modeled out an average values monthly of the photosynthetic fraction from imagery. And you can see that this does 2 things. This gives us real understanding of seasonal dynamics. We can see there in between 2011 and 2013, the Cowpam supersite, which is this is the core plot at that supersite, encountered some pretty wet years. You can see the impact of that. The area was impacted by fire in 2014. You can see that, and then you can see it coming back to a more steady state. This tooling didn't exist when we started out our network, but it certainly revolutionizes how we can understand those trends. And one of the beauties of it too is we can go back in time, and indeed this graph does. Half of this graph precedes TERN because we've got a rich satellite archive to go back on. Here's another way, a really insightful way of expressing very similar information. You can see how the photosynthetic fraction at this site varies through time. And what you can pick up from this pretty quickly is the site's slightly skewed to dry summers, more rain in winter, more growth in winter. But you can see some wetter years there, and you can see that fire I spoke of as well. Provides a way of analysing those sites that we've never had access to before. Yep. And LiDAR, I think, is another tool that we now have access to, and we're starting to implement operationally throughout TERN, that provides us with information that will enable us to assess change in vegetation structure at a scale, a speed, and accuracy that we've never had the availability to before. And someone in the chat touched on this, but citizen science has been around for a long, long time. But I think the way we implement citizen science has changed even over that last decade or so, where citizen scientists provide the opportunity for us to obtain more observations in more places much more frequently. But historically, it's had a bit of a bad rap about the quality of the data collected. And I think with increased training programs, apps to enable people to use that information, easily submit that data, provide guidance as they're collecting that data. I think citizen science is becoming rapidly a really useful tool to help us collect more information more frequently, to help answer those change through time questions that we're increasingly being challenged by. And then the other thing we have is pre-existing data and baselines, and these examples here are from our AusPlots network, but we have a baseline that have been collected over many years that we're able to compare against. But also we have a really rich sample library that researchers can use. And so we have like soil biological samples that you can look at the biological composition of those soils through time, or we have vegetation samples, herbarium samples, and traditional soil samples as well, all of which enable you to understand a baseline condition that was some time ago and then collect similar information now to understand change. Transects provide an opportunity to understand how particular aspects of the environment change through most commonly climatic or rainfall seasonality, temperature gradients, so along gradients, and certainly often placed in areas where we expect change. And one of the beauties of investigating things in this way is when you're looking at a future climate scenario along a transect like this, there are locations along the transect that are already an expression of the environment we're likely to experience in 50 or 100 years. And so substituting space for time in this kind of way enables us to understand change in a much more rapid time frame than waiting for it to play out and then just simply measuring it. The point I want to make by this slide is, I know I only showed one transect. TERN has many transects now, but we also have a network of sites that you can essentially choose your own adventure. You can make your own transect by intersecting the locations of our plots compared to modeled variables of interest, and then use that information to, essentially construct your own transect. You can see here there's essentially a transect from Darwin to Adelaide. There's many others there, depending on the question and what exactly you're investigating. There's a whole infrastructure here that's enabling you to utilize and answer the questions that you're keen to do. In regards to doing site experiments at locations where we already collect plots, and I think Hannah, you touched on this, we're keen to avoid actually sampling within our plots. And I've got a diagram to show these. We're really keen for you to sample adjacent to our plots, because then you enable yourself to tap into a huge historical wealth of data and samples, and often provide yourself with the baseline or the control from which you can judge your experiment. This is just a diagrammatic sample of the site at our Kaplan super site. And you can see the square made out of the little dots right in the center of the screen. That's actually what we would call our core plot at the super site. And then the other ones scattered around are actually other plots we've got in the same kind of environment. But you can see there's plenty of opportunities, and I've picked out sort of two communities here, a dune crest community in the sort of orangey color, and a dune swale mallee community in the sort of bluey greeny color. You can imagine those, you could establish plots with different manipulations in the same vicinity that's essentially sampling the same conditions, etc., that the plot we've already established, and tap into that historical depth of data and samples. By tapping into our network, you can benefit from the frequent information that our ecosystem process sites collect, and that's primarily around atmospheric fluxes and how the atmosphere interacts with the biosphere throughout many of Australia's major vegetation groups. We have, and you saw a map of this earlier, but we have now 1,000 ecosystem surveillance sites, all collected throughout all of Australia's major vegetation groups, all of the terrestrial ones, collect mangroves. So right across, basically across the continent, we have many examples of the same environment monitored, so there's pre-existing baselines. But then there's a huge archive now exceeding about 250,000 different samples based here in Adelaide that you can just tap into and use too. That's infrastructure that we enable researchers to use. And depending on what you want to do with them, that gives you some insight into historical conditions. Excuse me. You get to access to TERN landscape sites, and the landscapes capability of have some really intensively studied sites to help us understand calibration validation of Earth observation data. The couple of photos on the left there, and really interesting modeled information of soil and landscape across the whole continent as well. And then now, and this is an activity we just only 6 months into making operational, but we have hundreds of plots now collected in Drainscape, and Drainscape is TERN's UAV capability that cross-cuts. It uses skills across all aspects of TERN to provide photogrammetric, multispectral, and LiDAR information at many of our plots. The real advantage of this is, whilst the information content's lower, you can't, for example, always detect individual species using these kind of techniques. Depending on what you want, you can collect information much more rapidly. So an hour flight, for example, will give us really accurate measures of photosynthetic vegetation cover that would have taken a field team of 2 to 4 a whole day to collect just that aspect of what we do. So the tools and the program allows us to understand change more rapidly. And then we'll also provide access to TERN and to choose environmental monitoring system of Australia. And so a whole suite of standard methods that if you want to roll out an experiment alongside of our plots or whatever, I'd really encourage that you go to this website, consider the methods there, and if they're suitable for the kinds of experiment you're anticipating using, or and then that data will be available. We would like you to make that data available to others so that everyone can benefit and your data becomes intercomparable with others right across the country as well. That's it for me. That's the key points I wanted to make. My email and phone number are there if anyone wants to follow up after the presentations. Thank you.
Absolutely fabulous, team. Thank you. If you'd like to put your screens back on and your microphones on, we can take a few questions. We've got some already banked up, and I've had an SMS from somebody. I might start with that one. Mark, this one I think is for you. Somebody comments about the Australian Wildlife Consultancy, AWC, which fences off large areas, and obviously that does do things like of manipulate whether or not invasive species can get in, etc. Is that of any use in the sort of things that you're talking about? Is the question.
It does. Beryl, it's telling me that I can't start my video because it's disabled by the host, which I think it is you.
Yeah.
So if you want to see me as well, somehow this is up to you. Not to worry. No, disembodied voice will do fine, surely. Oh, here we go. Start my video. Ping.
Oh, I did it. Perfect.
Am I on? I am on. Yes. Okay. Well, look, they're interesting. They are a lot of things in parallel. So they're mostly targeted at exclusion of foxes and cats and particular predators. And then, of course, that has cascading consequences for what sorts of animals are there and for the vegetation and so forth. So it's certainly true that anywhere that you manipulate something about an ecosystem and it's and there is a control outside, then you have a comparison. I would say those are complicated because there's a lot of things changing, but they're certainly valuable. I'm not actually. I know that the different groups who've built these fence systems do some monitoring inside. I'm not sure how strong the monitoring is outside for the control. Be interested to know. And also, is it integrated at all? Are we using sort of common methods, between different exclosures, the data available to TERN, etc. So there's certainly some opportunity in there. It'd be good to find the ways to make best use of it.
Okay. I'll give you a rest for a second, and then I'll come back to you with the question that Sean has asked. But I'll go to Hannah now, because Bagal from Ethiopia has asked a question, and thank you for your talk, about considering diverse plant species with dynamic climate conditions is critical. Could you explain the factors that cause grasses and herbs to behave differently from trees? Is it physiological or other traits? Because I remember you saying that.
Thanks, Bagale. It's nice to see you in this webinar chat. It's great that you could join. So it's actually the shrubs and grasses that were behaving more consistently there, and I kind of sneakily used the word more consistently sensitive because the trees generally seem to be a bit more resilient to the temperature and water changes that we've inflicted on them than the other functional groups. But we think that we see a clearer signal for shrubs and grasses because they do more similar things within those functional groups. Whereas what we see for forbs is that they're actually extremely sensitive both to temperature and to water availability, but in very, very diverse ways. And we're still thinking about how to encapsulate that, trying to pin down what components of physiology or traits are responsible for those diverse behaviors in a set of 8 forb species, from an experiment of 32 species total. That's challenging to do. And we have more experiments in the pipeline and some field-based monitoring that might help to capture some more floristic diversity in the forbs and unpick that. So yeah, we sort of, I hope that sort of makes sense. It's not that forbs aren't sensitive to climate. They're in fact very sensitive to both temperature and water, but in really diverse ways within that functional group. So there's a lot more work to be done there to understand why certain species respond in the ways that they do for that functional type.
That's good. Now, I've got a question that's going to come to Mark from Sean, then I've got one for Ben, and then I'm going to go to Eddie's, okay? So, Mark, the one for you is on topography, such as elevation, slope, aspect, and constraining the realised niche of plant species in the context of dynamic changes in global and regional climate. Do you want to comment on anything there?
Absolutely. The only problem will be to stop me. Look, there's lots of interesting issues in that. Let me just mention a few of them. So first, as far as elevation, one of the slightly interesting things actually is that the great majority of the transplant experiments that across boundaries that we have are from higher to lower elevation, across temperature boundaries, are from higher to lower elevation up mountain systems, not actually with latitude. And you can kind of understand why, because the sites are closer, and so it's kind of more feasible to actually do the experiments. But you do wonder how much that's going to translate to the longer, slower gradients of temperature with latitude. Secondly, more generally with elevation, elevation and the inferred impact of elevation for temperature is in nearly all the species distribution models that have been built. Some of them also try to use slope and aspect, but the limitation there is that you have to be more confident about exactly where your specimen was collected from. So if you look at the big dots, Okay, you want me to shut up? Sorry, Bill. Okay.
We might
Can I
have
please
to
add something to that?
very quickly, Hannah.
Yeah, just to say that geographically distributed long-term monitoring plots are great for testing these questions because obviously we have lots of species turnover across latitude and elevation, but we also have species that remain in common. And if we have long-term monitoring data, we can look at changes in the dynamics of a species through time at different points in a landscape. So we can structure the analysis based on different components of topography. Obviously not as perfectly controlled as a carefully designed experiment would be, but we can get at some of those questions in ways that are possible with the data that we already have on hand.
Perfect. Thanks, Hannah. And Ben, question from Warwick about drone LiDAR data and whether there's a repository of accepted cited analysis methods for example, vegetation density, etc., that's used by TERN. Quick answer?
Yeah, quick answer, no. Slightly longer answer is we're desperately working on it. This program's only 6 months old and we've really got the collection through the storage routine now. And over the next few years, we'll be working really hard to make that raw data available and then hopefully analyze data as well.
And the next question I actually wanted you to all tackle, but maybe Ben, since you were just talking, you can start. It's from Eddie, and it's about scalability of experimental design, and it highlights, fauna versus flora, and slow growing versus fast growing, and so on. And do we have scalability built into our methods that you mentioned?
Yeah, I mean, I suspect both Mark and Hannah are probably more qualified to answer this than I am, but certainly different aspects of the environment vary at different rates. And so your experimental design is absolutely critical. How that fits into a background of long-term monitoring using fairly standard protocols gets down to exactly what you're looking at, I think.
Any extra from Mark?
Yeah, absolutely. I mean, it's a very absolutely scalability is an issue. It's a very, very general problem in ecology that usually a PhD or a research grant has 3 years. And it's as if you were trying to study chemistry and only be able to observe, 1/100th of the process happening. So it's a very important thing for ecology to start to get wider spread geographically, longer timescales to put these short experiments against the background of.
Right. And I'm going to skip the ability for Hannah to also comment, as I thank everyone again for coming along today. We always like to finish up fairly nicely on time. So we do have more webinars coming up. So in May, we're going to be looking at invasive species versus threatened species. We already have our speakers lined up for that one. It's going to be very good. So we look forward to seeing you in there. And just quickly chat. For those that haven't had their questions answered, we will take them offline and come back to you, because I see some are just coming in. But thank you very much, everyone. And just get my thing to go on to emphasize the thank you. And it was great to see international people here, because I think Mark suggested that some of these things are going to be best done at macro scale across countries. So we might come back to you later. Okay, see you all next time. Bye-bye.